In vitro models are needed because we want to study cellular interactions in the urethra, first of all in the normal urethra and then in the diseased one. We are studying diseased conditions in urethra by mimicking the inflammatory conditions, since several reasons might cause inflammation over there, diseases or trauma, for example. We are inducing the inflammation by adding inflammatory factors into the model. We can use, for example, interferon gamma or interleukins. We can also include inflammatory cells there in the model, or bacterial endotoxins. Why inflammation is important is that it is inducing fibrosis in the urethra. And that way there might be this kind of strictures in the urethra, so that the urethra lumen is narrowing. And fibrosis is causing that. If those factors that we are including in the model aren't inducing the fibrosis strongly enough, we can of course also modulate the bioinks what we are using in bioprinting so that they are stiffer to mimic the fibrosis. So fibrosis is inducing stiffness of the tissue. What we can also do, since we are utilizing bioprinting, we can really print the model so that some parts of the urethra are more strict and narrow, so that we are modeling also the strictures over there. Urethra is for urination. So we are mimicking urination over there. So periodic flow, periodic stretching of the urethra will be utilized over there to mimic the mechanical stimulation. In the lumen, we can also utilize artificial urine to see what is the effect of artificial urine to the composition, to the cellular functions of the urethra.